Okay, so we're going to continue with our understanding of ATP, which is adenosine triphosphate. ATP is used, like we talked about before, as the energy product and used in the cell. It's a primary energy currency used by all cells, and at some point, it is composed of ribose, which is a 5-carbon sugar, along with adenine, chain of 3 phosphates, which is the key to energy storage, bonds that are unstable and release energy when broken, ADP, which is adenosine diphosphate, 2 phosphates. And these 2 phosphates are the pre step to becoming the triphosphate, right? Remember that tri just means the 3 phosphate groups. So ADP is 2 phosphates, as we see in AMP being the adenosine monophosphate, which equals that 1 phosphate, which is the lowest energy form of our ATP, ADP, and lastly being our AMP. Now, molecules, energy bond shown here is what's being used to create that form of energy when we're talking about the usage within the body. So the ATP cycle is dealing with the ATP hydrolysis that drives the endergonic reactions. Remember how that was in our first section. And those coupled reactions result in the net negative G, which is that force, that work that we need, right? So exergonic and spontaneous is what this part of the hydrolysis endergonic reactions are. Now, the ATP is not suitable for long-term energy storage. We see that these are found in the phosphate bonds that are too unstable. So these are too unstable to be found for long-term storage. We know that fats and carbohydrates are better for storage of this energy, and cells store only a few seconds worth of ATP, just because how reactive those phosphate groups and those bonds are easily broken. So with that, we can go into our ATP cycle and how these inorganic to ADP and cyclical change all over again. We know that the inorganic allowing that phosphate group as phosphate, and as we break that phosphate group off, we now have the release of energy come back to the ADP, right? So that P energy, as we eat foods, we're going to now add phosphates back into our diet. So enzymes are needed because they are needed for the biological catalysts that are happening within our body. Remember, catalysts are things that are going to speed up reactions. And most enzymes themselves are proteins. And some themselves are also RNA. Remember, RNA is a single strand of those nucleic acids. And the shape of the enzyme stabilizes a temporary association between substrates. So enzymes not only are changed or consumed in the reaction, we also see there in other forms of this same reaction, example being the carbonic anhydrase. With this anhydrase, we see that the 200 molecules of carbon acid per hour are made without an enzyme. We also see 600,000 molecules are formed per second with enzyme as well. So this carbonic anhydrase can be seen in how it's formed, with our product being that carbonic anhydrase, which is our hydrogen group along with our added oxygen molecule. The enzyme binding is important to sites in which the substrate is complex, in which the binding now initiates something else to happen within the cell. This active site is where we see pockets or clefts for substrate binding, and they are forms of enzyme substrate complexes that we see in many different forms. So the active site involves a small number of key residues that actually bind to the substrates. Now, there's a precise fit of substrate into the active site that we see in this image here and here. And this applies to stress in a form of to distort the fit. This is an induced fit based off of the applied stress or that distortion. Now, the rest of the protein structure is needed to maintain these residues in position for the combining and the active sites for these enzymes to bind to. With our understanding of the substrate and the binding and active sites, we want to look at it in a different way in our figure 6.1. We can see that there are the substrate bonds shown here and forming an enzyme substrate complex. And we're going to see the binding of the substrate enzyme that places a stress on the glucose fructose bond, and the bond then breaks after that. After the products are released and the enzyme is free to bind to another substrate. From here this then forms enzymes, and these enzymes may be suspended in the cytoplasm or attached to the cell membranes and organelles. The multi-enzyme complexes are subunits that work together to form molecular machines. These machines themselves are the product that can be delivered easily to the next enzyme. The unwanted side reactions are prevented from happening because of the activation sites and the needed puzzle piece conformation within those active sites. So all reactions can be controlled as a unit based off of these substrates, the active sites, and the enzyme itself. The binding can be shown here where we're understanding these active sites once again, becoming the substrate complex substrates. Here's another way of looking at this in a more, so gives us different transformations. So we know the plus that occurs with that electron start this reaction within the proteins. So creating that transmembrane protein and those different subunits to open. And we know that proteins make up genes, right? So if these proteins are making up genes, then they're doing a lot with how genes are being upregulated and downregulated when it comes to why enzymes and catalysts are important. So with non-protein enzymes, these are ribozymes. In 1981, they were discovered that certain reactions catalyzed in cells by RNA molecules themselves. So two kinds happen in which we saw that the intramolecular catalysis happens, and then it catalyzes the reaction on the RNA molecule itself. And then an intermolecule catalysis, or the catalyst, excuse me, is where we see the RNA acts on another molecule. Within this, the ribosome at the bottom here shows that there is going to be the ribozyme, then the RNA message. After the ribozyme is introduced, we're going to see the cut and cleavage of the RNA message based off of that, what was communicated. Now, this is just an example, right? We know that enzymes also function in the form of rates, and the enzyme catalyzed reactions depend on the concentrations of substrate and enzymes. So any chemical or physical condition that affects these enzymes are three-dimensionally shaped and can change the rate. So when these dimensions are changed, these are based off of the change and denaturing function of proteins that we see with optimum temperature. So too high of a temperature will make these different three-dimensional shapes change, right? And denature and pretty much fall apart. When they're cold, most of the proteins then come together. So the optimum temperature is right in the middle in this form of homeostasis, like almost everything we talk about here in biology. The other thing is the optimum pH. pH is dealing with the alkaline and acidity that happens within cells, and these proteins need to be in a nice, comfortable, neutral pH. These enzyme environments shown here, once again, you can see a temperature reaction on the x-axis 0 to about 5 reactions at about 40 here. So 40, and the optimum is a little higher, over 70 degrees Celsius. So rates with that for pepsin, we see that at attic, and the optimal pH for trypsin is going to be 6.5 in reactions. So it's important to understand that these different reactions need different environmental cues. We also see inhibitors are part of this concept, where inhibitors are substances that bind to these enzymes and decrease its activity. So the competitive inhibitor are where we see this competition for that substrate and active sites in which they bind. So the non-competitive inhibitor that we see here binds to the enzyme at a site other than the non-active site, the active site, excuse me. So this causes shape change, and that shown here is where we see the substrates, and it normally binds to that active site and releases a product. But within our inhibition, we see and nothing happens, right? Because it's inhibiting the process of this enzymatic release and creating of products. Another example of this in this figure, you can see the same idea of this inhibitor. Blocking that competitive inhibitor interferes with active site of the enzyme so the substrate cannot bind to it. We also see that inhibitor changes shape of the enzyme so it cannot bind to the substrate. So these are both inhibiting products to be made by the enzyme. Now, we talked about catalysts, and now we have inhibitors. The allosteric enzymes themselves are enzymes that exist in active and inactive forms. So most competitive inhibitors bind to the allosteric site, and these chemicals are then switched on or off. Within the allosteric inhibitor, they bind to the allosteric site and reduce enzyme activity. With the allosteric activity, the binding at that site increases enzyme activity. So we have an inhibitor and then an activator. These working shown here, where we see the allosteric inhibiting it by having a substrate not being able to bind inhibitor on the allosteric site, which is on the other end of that enzyme. The altered active site wasn't there, but to move it back to a place where it's activation allosteric component. Now, the metabolism happens within all organisms, but the total, this is defined by the total of all chemical reactions that are carried out by that organism or an organism. So the anabolic reactions themselves are anabolism, and we see that this is a form of expended energy to build up molecules. And the catabolic reaction is where we see the catabolism. And with this catabolism, we see that this harvests the form of energy by breaking down molecules and those bonds that are found within foods. So glucose metabolism is what we see in this image, a form of intake, and disaccharides as well, the form different forms of sugars with glucose and fructose. Disaccharides are just two sucrose. Something happens in storage in form of glycogen, and this distribution and is utilized using the free glycogen that we find within the muscle cells. Within these biochemical pathways, the reactions occur in a complex and it happens in many different organelles, but in data phosphorylation that happens with the TCA cycle and producing that ATP. The product of one reaction is the substrate then has a domino effect for many other things to happen. So many steps take place in specific organelles for the specific ability for that specific cell. With these biochemical pathway examples, here's another example showing you that the of our polar heads and tails. The enzyme 1, intermediate substrate, the end product. Now, this is a cascading effect of that biochemical pathway example as shown before, or some of these pathways have to happen first before another one can be initiated. So also have these feedback inhibition. So the end product of pathway increases in concentration as it is synthesized. So more product increases probability that it binds to an allosteric site on an enzyme in the pathway and causes it to change so it cannot bind to the normal substrates. Now, this shuts down the pathway so raw materials and energy are not wasted. So remember that allosteric site is either blocking or producing a form of a reaction to that enzyme. And with the feedback inhibition example, we can see here that the initial substrates, the end product here, right? So the end product cannot be by the allosteric site on the other end of that enzyme. So with that being said, that is the end of this chapter. If you have any further questions, once again, please feel free to reach out and email me. Everything should be online within your syllabus. But if there's anything else that I can help and be assistance with, please let me know. That being said, see you in the next video.